While viruses are interesting for lots of reasons, of course, the primary reason they get our attention is because they cause disease. Viruses cause disease primarily by killing cells. Viruses infect cells, cells die, and whatever function those cells supplied is lost, and that loss constitutes symptoms. In addition, viruses cause disease because of the immune response that is mounted against the infection. This doesn't necessarily imply that one's immune system is too active. This is just the occasional but inevitable outcome of the way the immune system does its job. Rarely, viral infections do the opposite of killing cells. That is, they infect cells and instead of killing them, they disrupt cell cycle in a way that a tumor develops. So the tumor is sort of secondary to whatever cell function disruption that the virus did. It's interacting with some protein in such a way and altering gene expression that you get a tumor, which can, depending on the kind of virus, can develop into a metastatic cancer. And then lastly, we need to talk about the special example of HIV. HIV causes disease by immune suppression, but this immune suppression is secondary to, first of all, the death of T cells, and secondly, altering function of various parts of the immune system. All right, let's talk a little bit about exposure and infection before we go any further. Here you see an infected person who's shedding virus. So the expression to shed virus just means virus is leaving this person's body in whatever way it does. And in the case of coronavirus, of course, as we've all heard, it leaves the body on droplets coming from the respiratory system. And in the same close vicinity, we have a susceptible person. Now, there are two outcomes for this susceptible person. One is that the virus coming from the infected person never contacts any cell in the susceptible person, and thus, even though the susceptible person has been exposed, they have not been infected. A second alternative is that one or more virus particles from the infected person actually makes contact with a cell in the upper respiratory tract of the susceptible person. So at that point, maybe or maybe not has the infection begun. So the virus inoculum has entered our person, our exposed person. The inoculum in a natural infection is generally thought to be pretty low. This is the sort of thing that it's a little hard to test experimentally. In the lab, of course, when you're doing an infection with a lab animal, you generally give a pretty big inoculum because you want the animal to get infected. But we know from experiments where you give very small amounts of inoculum that it's not uncommon that with a small, just a couple of what the equivalent of just a couple of virus particles, that the virus just may not successfully complete one infectious cycle. And we call this an abortive infection. So the virus has entered one cell, has sort of tried to replicate, but for one reason or another, just maybe it viruses are sort of sloppy actually. There's just a lot of them and they're successful because there are a lot of them. But abortive infections are not uncommon. And then of course there is what we call the innate immune response. We're going to talk a little bit more about immunity in a subsequent lecture. But for one reason or another, the infection might be abortive. And in that case, there will be no detectable record of this event. This person will never test positive, no antibody has been produced. You won't know what this is. The other alternative is that the virus completes the infectious cycle in the first cell, progeny is successfully produced, and they infect other cells. This person is infected. Now, how long it is before there is some sort of positive test, of course, depends on the virus. In the case of the coronavirus, it would probably be about a day. Again, this is a little new. We don't have all the data that we will say 6 months from now when we're able to, we're able to stop being in emergency mode and be able to start actually dealing with the information we're getting.
So the person is infected, but are they sick? Now, that's not a straightforward question. It depends a lot on the virus. So let's first take the example of something like rabies virus infection over here. For rabies virus infection, I shouldn't say almost everyone who is infected with rabies virus who develops a symptomatic infection, they're all going to die. Rabies virus is among the most virulent viruses known. The outcome, there really is no treatment. It's maybe a few odds and ends of people who survive, but for the most part, you get infected, you develop symptoms, you die. On the other hand, we've got something like a common cold virus, and the official name for common cold viruses are rhinoviruses. Rhino meaning nose, because it infects your upper respiratory tract. So a rhinovirus infection is generally pretty mild. So down here, it's pretty mild. There's a big range of symptoms, but nobody gets terribly, terribly sick. Influenza A virus infection, giving you the disease influenza, a bigger range. Some people have a completely asymptomatic infection. Some people have very serious infections. And as you've probably already heard, because coronavirus gets compared to influenza A virus infections a lot, that roughly 20 to 30 million Americans die every year of influenza A virus infections, but there's a whole lot more infections than that. And then a fourth thing I put on this graph is HIV infection. So AIDS, which is the disease caused by HIV, AIDS presentation is quite broad as well. There are a small number of people who seem to not get sick at all. Most people certainly, now this would be in the absence of treatment, right? With treatment, of course, the whole curve moves to the left. But in the absence of treatment, most people are going to be very sick and will ultimately die, and some won't.
So, what causes the curve in some cases to be so very broad? Well, much of the time it is about, do this, yes, it's about the host. Um, Host variation, age, nutritional status, the presence of other illnesses, what's the status of your immune system, and a lot of that is in turn related to age because our immune system changes a lot as we grow old. What are the bacteria are in the individual? This is particularly the case for viruses that infect the gastrointestinal tract. The microbiome, or all the other bacteria that are in your inside of you, can really influence the course of disease. This has been shown experimentally in the case of poliovirus. Of course, the other part of an infection, right? Part of it's the host, part of it's the virus. And so, what variation is there in the virus? In some cases, there are multiple strains circulating for a given virus. Now, we just don't know the status for coronavirus yet. People are measuring and analyzing lots and lots of isolates. It doesn't seem at the time of this recording that there are significant strain variations, but that is something that could be developing. The route of entry. How are you exposed? In the case of coronavirus, again, you generally are exposed through the upper respiratory tract, but other viruses you are exposed differently. In the case of HIV, you can be exposed through sexual exposure, through intravenous drug use, through a blood transfusion. Blood transfusion exposure by blood transfusion is not the case now, but certainly in the 80s, many people were infected by blood transfusion. What's the size of the inoculum? Are you exposed to a relatively small dose, as we described in the previous slide, or a much bigger inoculum?
Many viruses exist as co-circulating variants, which are each stable. A good example of this are rhinoviruses, which cause a really large percentage of common colds. There are about 100 serotypes of rhinoviruses. Now, traditionally, serotypes were defined by whether an immune response to one type could neutralize or prevent infection by another serotype. This gets tested in tissue culture generally. These days, serotypes are sometimes defined instead by genome differences or percent of genome differences. In these cases, there has to be some definition of a functional sequence or an exact percentage of difference or a location where there's a difference that's required to categorize a variant as a different serotype. That is, just because one virus strain is different in sequence from another strain doesn't mean that it qualifies as a serotype. Now the question remains why some viruses, like rhinoviruses, exist as multiple stable serotypes, while others, like measles, exist as just one. It's not that there's no regional variation between strains, but the variation in measles is small enough that an immune response to one strain still will neutralize another. And there's no answer to that. It's just that evolution has produced different outcomes for different viruses.